Okay, so we are going to start chapter 9 today, and this chapter is all in genetics. I like this chapter. It's a pretty fun, straightforward chapter. So what we're going to learn about in this chapter is everything about heredity. We're going to learn about phenotypes, which is how we see things in your body, why your eyes are blue, why your hair is a certain color, why you have the certain features of your body that you have. So starting off, let's go back in history. We have a bunch of monks here. And the main one we're going to talk about, and I always find this picture funny, but the main one we're going to talk about is Gregory Mendel, who is right there. And this picture is just, everyone is everywhere. You got this guy here, he's looking that way. You got this guy here, he's looking that way. A lot of people are looking to their left or right. It's just awkward. And then there's Gregory Mendel that's holding a plant in his hand. But anyway, so section 1 is all about Mendel. Now, he was a monk. He worked or lived in a monastery. And monks were very religious people. And along with praying and, doing things like that, they had a lot of time on their hands as well. So a lot of them started to dive into the sciences. So when Gregory Mendel entered the monastery, his whole job was to tend the garden. And this is actually his garden in Austria. It's still kept to this day just because of all the contributions to science that he made. So he researched heredity. In other words, the transfer of characteristics from parents to offspring. And he did pea plants. He worked with pea plants a lot. Why pea plants? Well, they can reproduce very quickly, and it's really easy to see all the characteristics of pea plants. So he was able to study them very easily because he had so many offspring that he was able to look at. He observed a lot of different characteristics on these pea plants, and we called these different things that he saw, we called them traits. So for color, he saw two different traits. He saw that some peas were green and some peas were yellow, just like there. But he also saw a bunch of other things. If you look at these two here, we have the bottom ones, which are smooth peas. Then we have the top ones that are wrinkled. So he studied those traits too. A lot of them right here, here's 4 of the 7. Plant height, flower position, because pea plants do have flowers. I know, you guys just see the pods when you eat them, or even just the peas, but you don't really see the flowers that are on the pea plants. He did the pod color, which we talked about already, the seed texture, the seed color, so what color the peas actually were, and then those flowers actually have a color too. Some of them are white, some of them are purple, which is kind of funny to think of, which you have a purple flower on a pea plant when the peas are either going to be green or yellow. But and then he also saw the pod appearance. So he looked at all these different things. And what he did was he messed with pollination of the pea plants as well. So how you can do different things with pea plants, guys, is you can either have the pea plants self-pollinate or cross-pollinate. Now there are different parts to that plant. There actually are female and male reproductive organs on that plant. The male part is called the anther, and the female plant is called the stigma. The stigma is kind of down the middle of the pea plant, and the anther, if we're looking at the flowers, it is the part that is slightly lateral to the stigma, and that anther is going to have the pollen attached to it. So we have the stigma and the anther. Now, if the pollen can get in through the top of the stigma, so it would have to go here and then drop right down the middle there, it'll travel through this tube all the way to the ovary where pretty much fertilization occurs. But we need to get the pollen from the anther to the stigma. Now, 2 different things we can do here. If it's the anther of this plant, and it goes to the stigma of the same plant, we call that self-pollination. It could also go from this anther here to the stigma of another plant. That is called cross-pollination. So that is one plant pollinating another plant. Sorry guys, bell. Between self-pollination and cross-pollination, self-pollination not going to give us a lot of genetic variation. If we do cross-pollination, we're going to get a little bit more genetic variation because we're using two completely different plants. And again, here's another picture. You can see the stigma a little bit better in this one. And again, those anthers are going to be surrounded with pollen on top. And here's just what we went over, guys. Self-pollination, stigma, anther, it's all one flower. And then we have cross-pollination. Here, watch. This is so cool. I made this animation. I'm proud of it, so enjoy it. But if you guys see there on the anther, I put some little pollens. Now, how does it get from one flower to the other? Well, the wind, yeah, that's a good one. But at the same time, we can't just have the anther just accidentally get it over to the other plant. So bees also play a big role in cross-pollination, where they pick up the pollen from one plant and they will take it over to the other. Now they don't literally, guys, go, oh, here, let me take your pollen and I'm going to take it over here for you. No. The pollen, if you look at the little spikes on it, they actually get caught on the bee, and the bee, without even knowing it, carries out cross-pollination for our plants. All right, so there is a bee just completely covered in pollen. You guys can see it all over it. And then once it leaves that plant, it's going to go to another one, and probably some of them pollen are going to get into the stigma of the other plant. So what Mendel did was he was able to transfer the pollen from one plant to another, and he was able to self-pollinate and cross-pollinate. And sometimes he wanted self-pollination to occur. Sometimes he did not. Sometimes he actually wanted to make sure it was going to cross-pollinate. So what he did was he would actually remove some of the anthers from some of the flowers to make sure, okay, that flower is unable to self-pollinate. And that way it would have to get an anther from another plant. Okay, so going back to our karyotypes from last chapter here. So I got all of our different chromosomes on here. So what's going to go on here, guys, is we're going to talk about different traits. Now you're going to get some of these characteristics from the chromosomes, and one of the characteristics is going to come from one chromosome, and the other characteristic is going to come from the other one. So in other words, you're going to have one from mom and you're going to have one from dad. One of them usually is going to overpower the other one. So dads might overpower moms, or moms might overpower dads for any particular trait. That's what we're going to focus on at the beginning. Come on. At the beginning of this chapter, we'll focus on that, just one gene dominating or one trait dominating another. But later on, we're going to see what happens if one actually doesn't dominate. But remember, guys, you're going to get one of these from mom and one of these from dad. So let's give names to these things. What we're going to say is each one of your chromosomes for any particular trait is going to have something called an allele. So A-L-L-E-L-E, an allele. Some of our alleles are going to be dominant. We represent these by capital letters. Some of our alleles are going to be recessive. We're going to represent these by lowercase letters. The dominant allele for right now, for our intents and purposes right now, our dominant allele is going to overpower the recessive allele. So if I go back, let's go back to our karyotype here. Let's say that, let's just going to use red for mom. In number 1, you get this red one from your mom. And we'll do blue for dad. So there is blue for dad. Okay. So if dad has a particular trait, and I'm just going to highlight this trait in black. There's a trait right there. I don't know what it's for. It doesn't matter. But that trait is going to be in the same position, the same area on the mom chromosome. All right. Why? Let's go back to last chapter. Do you remember when we went over crossing over during prophase 1 of meiosis 1? He just talked really fast. Remember when we talked about crossing over during prophase 1 of meiosis 1? Some of these are going to flip-flop. So we want to make sure those traits are in the same area so that when they flip-flop here, that this trait is going to go to dad and that trait is going to go to the mom chromosome. So that way the kid gets that trait, whatever that trait is. We don't want the kid to not have a trait for like eye color or something like that, you know. We need eye color to protect our eyes from the sun. Anyway, so if the mom's allele, let's say, is capital, and the dad's allele is, we'll say it's recessive, so lowercase. What this tells us is that the mom trait here with the big R, that mom trait is going to be expressed or seen, and the dad trait is going to be masked. Okay, so that dad trait will not be seen. And you can see here, guys, they highlighted all of them. And you can see some of the different traits there, but the colors are consistent going across from one to the other. Okay. Here is a haploid cell. And just so you know, like, you know, for the haploid cells, you're going to get one of these from mom, one of these from dad. And that's why we are going to have 2 alleles for each one of our characteristics in our body. Now, what do these alleles make up? Well, the alleles are going to make up something called the genotype. So the genotype is the alleles that you get for any particular trait from your mom and your dad. Your genotype is going to consist of 2 alleles. So your genotype always, always, always is going to be made up of 2 alleles. Where do you get these alleles from? One from mom, one from dad. So genotype made up of 2 alleles, one from your mom, one from your dad. Okay? So we have 3 different possible variations for genotypes with respect to those 2 alleles. Again, the one you get from your mom and the one you get from your dad. Now, some prefixes I want to go over with you guys. Homo and hetero. So homo or homeo means the same. If we have it in this example here, I have a big R and a big R. So I have 2 dominant alleles grouped together, and that makes up your genotype. So here's our first genotype. We call this one homozygous dominant. The homozygous tells us that the letters are going to be the same. They're either both capital or they're both lowercase. The dominant tells us that they are both capital. So that's our first one. Our second one is going to be homozygous recessive. So homozygous, once again, tells us we have 2 of the same type of letter, whether capital or lowercase. Recessive tells us that they are both going to be lowercase. And then our last one, big R, little r, this is heterozygous. The prefix hetero means different. So it's 2 letters that are different. We do not need a dominant or recessive after heterozygous because there's only one variation where we can have a capital and a lower, or different, letters. So heterozygous is 1 capital, 1 lowercase. So there are your genotypes, and those are your names for your genotypes. Make sure you know these because we are going to be using these terms a lot. Homozygous dominant, homozygous recessive, and heterozygous. We're going to be using these a heck of a lot. So you need to know what they correspond to. That homozygous dominant is big, big, homozygous recessive is little, little, and heterozygous is a capital with a lowercase. What they determine is the phenotype. All right, the phenotype is what we can see. So let's do a little example of this one here. So this little boy has a condition called cystic fibrosis. Cystic fibrosis. Oops. So he has to get treatments for his lungs. Why? Well, if you have cystic fibrosis, what happens is you have a very thick mucus in your lungs. And that mucus sometimes becomes more mucusy than other times, I guess we could say. Their life expectancy is usually in their 30s, people with cystic fibrosis, and it is caused by a homozygous recessive genotype. So this is a phenotype. Cystic fibrosis is a phenotype that is caused by a genotype. So this is a condition. If you are, I'm just going to use R for an example, guys. If you are big R for your genotype, in other words, if you're homozygous dominant, you don't have cystic fibrosis. If you are heterozygous, big R, little r, you don't have it. But if you are little, you do have the phenotype of cystic fibrosis. And cystic fibrosis is on chromosome number 7 there. So that is the chromosome that has this condition on it. So if you have, for one of the traits on chromosome 7, because there's thousands on there, if you have a recessive allele on the one that your dad gave you and a recessive allele that one your mom gave you, you will have cystic fibrosis. Okay, so we'll stop here, guys. We'll do some more examples tomorrow. Have a good rest of your day.